All right, everyone. So I'm excited to kick off the winter quarter of the GBSB seminar by introducing Alexander Hoffman. He is the Thomas M. Asher Professor of Microbiology and Immunology at UCLA. But before he left for UCLA, he was here at UCSD. I'll say he is, both a great researcher, but also a serial sort of leader and I guess mover of things. He has founded and is directing the Institute for Quantitative and Computational Biosciences at QC Bio, but he also was the founder here of the San Diego Center for Systems Biology, and also co-founded the Biocircuits Institute, and was very influential in further sort of developing the bioinformatics and systems biology graduate program. So I think we're really excited to have him come back and visit and tell us what he's been up to.
Thanks so much, Hannah, and thank you for the invitation. It's really great to be back, actually. It's super fun, and it's great to see familiar faces and meet new people and get updates and be here with this great weather when, we've just had crazy rainstorms in LA for the last few days. So it's great to be back because, yes, both the San Diego Center for Systems Biology and now QC Bio have been a big part of my life. And, as life and time flies and time moves, and one way to monitor how time goes is sort of by looking at pictures like this, where I'm Tracy and I are walking along La Jolla shores, or, La Jolla with our little kid. And then 10 years later, we're cycling to San Diego from LA. And, if you saw me wandering around campus last summer, that was because I was on a cycling trip. So, but let me dive into some science. And so in a seminar like this, this sort of graphic or figure really doesn't need much introduction, right? This is a single-cell RNA-seq analysis. And it is looking at a mixed populations of cells. And the power of single-cell RNA-seq is that numerous RNAs, numerous analytes are being measured, and their combination describes the identity of cells. And this is really, that has revolutionized almost all aspects of biology, certainly developmental biology, and certainly immunology. And it has also affected the way we do science in my lab. And we're interested in macrophages. And one aspect which you find in many clinical studies, just like this one's on COVID, is that there are a large number of actually different types of macrophage populations. Look at all of these here are macrophage populations, and they are distinguished by the single-cell RNA-seq. And yet the distinction isn't really very clear by this UMAP analysis. They're all overlapping. Somehow the colors are presented here as if they're different, and there is some clustering that underlies that. But even this really super powerful visualization doesn't really distinguish them properly. And so we're wondering why that is and what's going on here. And particularly in my life here at San Diego, we focus not so much on bioinformatics, but on dynamical systems, on the dynamics of signaling. And so with that background, I'm reminded, and I'd like to remind you, that the steady state of a cell, or any dynamical system, and a cell is a dynamical system, is not just a function of the abundances of the proteins or the mRNAs, but also the fluxes. That sort of fundamental dynamical systems understanding, or another way to describe it is that's really the challenge of creating artificial life. We can make all the different components, put them all into a lipid bilayer, but then somebody has to give it this magical push, in order to get the dynamics going, in order for life actually to happen. And if we're measuring just the abundances with single-cell RNA-seq, we're missing information in order to describe the cell state. And of course, that's a technological hurdle. How do we measure dynamics? And so I'd like to address in this talk, our attempts to do so, to try initial attempts to measure dynamics and to then infer cell identities based on the dynamic measurements. So that's the big picture. So before we get into, before I can tell you a little bit about the research, I need to have some introduction about macrophages, because not everybody is familiar with macrophages. Macrophages are phenomenal cells, amazing cells that show a high degree of stimulus response capability and specificity. They have dozens of receptors recognizing hundreds of ligands and deploy different types of functions depending on what stimulus they were stimulated with. They do so in a manner that is context dependent, depending on where they are in the body, what microenvironment is present. They will deploy different functions. And they can integrate prior stimulation into their responses and their functionality. They have memory, depending on what they have seen in the past. So those three hallmarks are hallmarks we recently described in an annual review. And so I'll go into each of these in a little bit, which makes them so variable. So the stimulus responsiveness is that they are able to recognize many different pathogen-associated molecular patterns and also damage-associated molecular patterns, and deploy different functions, whether they're cell-intrinsic defenses against pathogens, or activating the local immune response by recruiting other cells to the site of infection, or activating systemically immunity and the adaptive immune response specifically. And each of these various functions are deployed in a ligand or stimulus-specific manner. And we know a lot about the pathways that do that, the molecular pathways that do that, and I don't need to go into that. But one mechanism, one key regulator of this is the transcription factor NF-kappa B. And so NF-kappa B was the focus of studies that are also had here at UCSD. NF-kappa B is a transcription factor that responds to all of these different receptors. And importantly, those receptors are not on-off switches for NF-kappa B, but they modulate the dynamics of NF-kappa B in a stimulus-specific manner, which leads to this rather, irreverent diagram here, or old school telephone receivers might be lost on the current generation, but there's amplitude modulated signaling. And so one question we asked, which is sort of an introduction to where I'm going to go with this, is if these are amplitude modulated signals, they are dynamic signals. And this represents a code, meaning that the dynamics of signaling that is encoded by these receptors and the molecular networks around it, and are decoded in the nucleus to then determine different functionalities. If this is a code, then there must be code words. There must be dynamical features that actually mean something. Otherwise, the term code really doesn't make much sense. You have to have code words if there's a code. The genetic code has code words called codons. And so we attempted to identify those dynamical features that are meaningful by generating a ton of data. And that actually was initiated by a graduate student here at UCLA, Brooks Taylor, UCSD, sorry, Brooks Taylor, who was a bioengineering grad student and then actually continued at UCLA with me, and then trained an MD PhD student, Adelaya. to generate a knock-in mouse that works really nicely with reporters, and Brooks generated an automatic image analysis pipeline so that we can extract NF-κB dynamics as a result. And the kind of data we get is like this. The live cell microscopy is shown here. Cells are moving around, but the image analysis can identify nuclear NF-κB quite well. If you focus here on a nucleus, you see NF-κB going in and out in response to a stimulus like TNF. But in response to a stimulus like LPS, NF-κB really remains in the nucleus and does not oscillate. So this is one example of the dynamics being stimulus specific. And by measuring hundreds of these cells, we can generate heat maps where the x-axis is time over 12 hours or so. And you see how, in this case, really every cell shows an oscillatory pattern, which is completely lost when you average the data. That's why single cell analysis is so important, right? And whereas with LPS, they do not oscillate. And so, so in this way, we could generate lots of data at different doses for different ligands, and ultimately, and now ask, are there any features, any dynamical features in these trajectories other than oscillations that that correlate with the stimulus? So, because if they correlate with the stimulus, then they're informative about the stimulus. And so one way to address this is to, first of all, generate a library of different types of dynamical features, for example, integrals or derivatives, or peak amplitudes, or peak time, or time of the peak, or durations, or oscillatory frequencies. These are all dynamical features that describe trajectories. And ask which ones of these dynamical trajectories correlate with the ligand. And so to do that, information theory is really the right tool. It's a correlation test that allows for non-monotonic relationships. And so where the stimulus is the source of the information and the destination of the nuclear target genes, and NF-κB is what is transferring that information. And so by correlating trajectories of NF-κB or different metrics, different dynamical features of NF-κB trajectories to the ligand, we can identify, we can measure the information content, and we can identify the most informative features. And so by doing that, we find that by looking for the most informative features, basically at about 7 features, the amount of information content that we can extract from this plateaus out. And so those 7 features, dynamical features, represent informative features about this data set and about the ligands. And so what are they? There are 2 early derivatives that really describe the speed of activation at this in this short time course here. There's the peak activation. There is another derivative that describes whether there is a post-induction repression event, which indicates the oscillatory behavior or non-oscillatory behavior. And then there's total activity, or the integral. There's how much total activity is early, how much of it is front-loaded, the early versus late activity. And then the total duration of the response is also another important dynamical feature. So we have 6 dynamical features that are informative about the stimulus that convey information to the nucleus. And when we submitted this to immunity, they felt that that code word somehow was a confusing term. And so we settled on the term signaling codons. Units of information in the signaling pathway, similar to the units of information in the genetic code that conveys information from one generation to the next. Here, the signaling codons convey information from the outside of the cell to the nucleus. So with 6 signaling codons, these are deployed stimulus specifically. Of course, there's a lot of variability. If we show the violin plots for TNF, you see there's lots of cells have, for example, high speed, but some are low. But you see a stimulus-specific deployment of the speed signaling codon. Oscillatory codon is very high for TNF and much less than LPS, for example, or the other PAMPs, and so forth. You see stimulus-specific deployment of these signaling codons. And so we now asked, but can we use these signaling codons or these code words as a way of learning the language of immune cells? It's a nice allegory or metaphor, and ask whether we can identify the stimulus based on the signaling codons alone. And what's shown here by this ensemble of decision tree classification scheme is that the F1 score is almost as good with just the 6 codons as for all features together. So they really do capture the information about each stimulus. And that's also shown here in the diagonal, the degree to which each ligand is properly identified. But in addition to that, this confusion matrix, of course, also shows where there's potential confusion. And we see, for example, high confusion between panther-CSK and LPS. And maybe that's not so bad because both of them are bacterial ligands, whereas the confusion to cytokine is much less, and the confusion to viral PAMP is also much less. So in some sense, it's the bacterial ligands that are less well distinguished from each other than the other categories. And so then we can combine bacterial ligands together, and we see that there's really good distinction. And this is just based on NF-kappa B dynamics, not considering any other pathways that might also be activated by these ligands, right? So the dynamics really do show a lot of information about the ligand. And then the question is, does this really matter? And so we bred the mouse into this reporter mouse into a mouse model for Sjögren's syndrome, an inflammatory disease that sporadically occurs. We isolate macrophages from that, and we examined the signaling codons, and we find that most signaling codons are deployed basically similarly well between the two, between the healthy and the Sjögren's syndrome macrophages. But the oscillatory codon is not distinguished. And also there is a problem in the deployment of this duration codon. So there are 2 specific dynamical features that are defective in this particular mouse model of Sjögren's syndrome. And as a result of that defective deployment, the level of confusion between TNF and LPS is much, much higher. That means the Sjögren's macrophages from this mouse model, I'm not talking about human Sjögren's patients, but from this particular mouse model, interpret a TNF signal as if it was endotoxin in terms of NF-kappa B dynamics, which means a completely, an exaggerated response, a confusion of what they're actually seeing. So we were pretty excited about this, and so let me stop at that point. But it basically says that signaling codons are a way of decomposing these very complex trajectories. And the 6 codons are sufficient to do that. They can capture all the information, and that information is physiologically and pathologically relevant. So signaling codons convey information about the stimulus, and misdeployment of these signaling codons, of a specific signaling codon may lead to a signal confusion disease. So that's about response specificity. And a subsequent question then is, well, if we have these 6 different signaling codons, how do the nuclear target genes distinguish between them, discriminate between them? How do they decode them? And actually, starting with Robin Lee in Suzanne Godet's lab, we have a pretty good understanding of this. And we then continued on the speed, on the amplitude codon or the speed codon. We continued on the duration codon, another, and then most recently the oscillatory codon, the one that we're pretty excited about, actually is not so much decoded by the, or distinguished by the poised immune response genes, but more by the latent enhancers and therefore the subsequent responses. And so the oscillatory signaling codon therefore plays a key role in determining the stimulus memory that I alluded to at the beginning. But that's really for a different talk. And so I wanted to go on and then talk about how context dependence of these dynamics, whether the context dependence, whether the dynamics of signaling and the response specificity is affected by different contexts. And so for that, we use the same experimental pipeline, but instead of just stimulating cells with different ligands, we previously conditioned these macrophages in a variety of different microenvironmental cytokines, type 1 and type 2 interferons, IL-10, IL-13, and IL-4. And so that led to even more data. In fact, about 68,000 cells totally, for which we have trajectories, so close to 7 million data points. And so in examining this, this is a correlation plot for 2 replicates, replicate 1 and replicate 2. And fortunately, we have good correlation between these trajectories, so the experimental pipeline is working. But what you can see here, by taking the M0 macrophages or interferon beta, preconditioned interferon gamma, they actually do not correlate anymore, which means that these microenvironmental cytokines affect signaling dramatically. In response to R848, not so much, but in response to TNF, a lot, and so forth. So there is a huge amount of information in this, but the first thing to notice is that polarization really does alter the stimulus responses. So, so we wanted to get at that in a little bit, and then used another machine learning tool, LSTM models, that do not require the identification of any features to do classification. And what we find here is that a particular stimulus in these data sets, in the M0 data sets, can be identified from the trajectories pretty easily, some better than others. But importantly, in response to different polarization, some stimuli are dramatically affected by the polarization state, whereas others, like R848, not so much. And here's an example for PAM3CSK. PAM3CSK is the one that is highly affected. If you look at the confusion matrix, in the M0 macrophage, there's some confusion of PAM3CSK along these other bacterial ligands, flagella and CPG, FSR1, LPS. But when but in the IL-10 preconditioned macrophages, PAM3CSK is also confused with poly-IC. And even more, and with, mostly with poly-IC. So, and the others. So you can see, you can really drill down how this signaling code is affected by polarization. And we also asked whether we can identify dynamical features that are informative, that drive this classification. And so for this, we actually limited our library of 918 features to just 71 that we knew were really uncorrelated, and to reduce this computational effort. And implemented this model, used separate validation and then testing to evaluate the performance. And what we find is that this this feature-based classification actually works at least as well as the featureless classification I described earlier. So, and that one reason for that is that identifying the features which involve multiple time points tends to reduce some of the technical noise that we have in our data sets. So, so with that, we can use this to figure out what is the loadings on these machine learning in this machine learning classifier to identify the most important features that have the highest loadings, and then remove one feature at a time and see how many we really need until classification performance drops. And there's about 18 that are needed. And with those 18 features, then work just as well as the original 71. So, and those 18 features actually describe if you will, signaling codons that could be combined into 7 or 8 different sort of overarching signaling codons. So, so using this approach, we can find now, we can track again confusion between different responses. So for example, here, we see that that whereas poly-IC is highly distinguishable from LPS. Oh, sorry, there's a high amount of confusion between for poly-IC and LPS. R848 remains identifiable, and there's very little confusion. So there is a lot of specificity here. So poly-IC is highly affected by interferon beta responses, and not interferon beta pre-stimulation. And so, whereas the M0 macrophages, you see that the poly-IC responses can be distinguished well from LPS responses. In interferon beta preconditioned macrophages, those trajectories are less distinguishable. And then we can drill down and ask, well, what are the features that are driving this? And so I just want to point out, for example, just this feature called integral, one that the early integral between a half-hour and 1 hour time point is very different for LPS versus poly-IC in M0 macrophages, but in the interferon beta pre-stimulated ones, that feature is much more similar, and that is what is causing this increased confusion. So what I've shown you is that the trajectories reveal the specificity of stimulus responses and how they are affected by polarization. But let's go back to the original question. How about the inverse? Can the trajectories also that are elicited by the stimuli, can they also reveal the specificity of the polarization states? Whether the cell states determined by polarizing cytokines, whether they can be distinguished, not based on single-cell RNA-seq, but those trajectories. And so how do you do dimensionality reduction on dynamical trajectory data? instead of PCA, there's something called functional PCA. Functional PCA uses a set of different dynamical curves and ascribes different loadings to those curves. And so implementing functional PCA to describe the trajectories that are in the data, we find, for example, that the different polarizing states can be distinguished with an F1 score of around 0.4, ranging from about 0.32 to 0.44. And of course, the trajectories have to be elicited by a stimulus. So when the trajectories are elicited by poly-IC, the polarization states are poorly distinguishable, 0.32. Whereas when the trajectories are elicited by PAM3CSK, then the polarization states can more easily be distinguishable. And so with this functional PCA, then we can put those on sort of a familiar UMAP visualization. Oops, this somehow got in the way here. But what I want to show is that when the trajectories are listed by PAM3CSK, then the different polarizing states do live in different areas of this UMAP plot. Whereas, and this is too bad, whereas if they elicit by poly-IC, which has a much lower F1 score, oops, then they're actually more on top of each other. So that was using a featureless dimensionality reduction. But we do have some features. We have these signaling codons. So now we can use those signaling codons in a feature-based dimensionality reduction. And we've now found that we can identify different cell states with actually a little bit better. The performance is a little bit better. PAM3CSK does much better. And even poly-IC does a little bit better. But still, they're very different. Or poly-IC does worse than PAM3CSK. And again, that is then can be put on a map. You see that poly-IC really does a poor job. The trajectories elicited by poly-IC are too similar between polarization states, so the polarization states can't be distinguished very well. Whereas in response to PAM3CSK, those trajectories are affected highly by the polarization states, and so the cells that are, for example, this is the IL-10 conditioned cells, can be distinguished very well from the M0 cells or or what's down here, the IL-4 cells and so forth. And so those are the two extreme examples, and then the other ones sort of fall somewhere in between, and I think the F1 score kind of indicates that. So let me summarize this part. Stimulus response specificity and also which dynamical features enable this stimulus response specificity depends on the polarization state. And the flip side is that the stimulus responses to some stimuli allow identification of the polarization state. And some, so PAM3CSK allows for identification of the polarization states, but the stimulus responses to poly-IC don't allow it very well. So that's with one analyte. Unlike single-cell RNA-seq, where I'm measuring 1,000 genes, I'm only measuring one over a time series. But it does indicate that cell identity can be defined by network dynamics. If only I could measure more analytes, not just one, but many, in a dynamic manner, right? So we're measuring NF-kappa B. NF-kappa B is in this network. There are many other transcription factors. If we had fluorescent reporters for them and we could multiplex fluorescent reporters ad nauseum, that would be phenomenal. And then we could have lots and lots of dynamic data. And maybe that's worth investing into. But let's think about the target genes. There's lots and lots of target genes that are activated. And of course, we do have single-cell RNA-seq, and we can measure all those target genes by single-cell RNA-seq and the responses to them. So that's the work of Catherine Chu, another MD PhD student who did amazing work, actually with Aditya Guru, who I was an undergraduate with him and made some great contributions. And they measured the responses in polarized macrophages, again, at a single time point to all the different stimuli. And what they find is that the responses are distinct, different clouds, and that distinction is different in M1 macrophages and M2 macrophages. So for example, CPG here is very easily distinguishable from LPS in M0 macrophages, but they begin to overlap in M1 macrophages. And the distinction also between poly-IC and interferon beta is less good in M2 macrophages than in M0 macrophages. So also gene expression responses are affected by polarization. Similarly, you can take macrophages that are not conditioned in defined polarizers, but in very complex environments in vivo, macrophages from healthy low-fat diet mice, or really old mice, or high-fat diet mice, macrophages extracted from them, stimulated with different ligands, show distinctions in their response specificity. So here, PAMP3-CSK and TNF responses are affected by age, and then also by high-fat diet, but in different ways. And so this is sort of introduction to this is its own study, which sort of attempts to use this method as a way of analyzing macrophage states based on a single time point. But we're interested in the dynamics, and from a single time point, we can't infer any dynamics. It's just a response at a single time point. And so Catherine then went on in more recent work to actually measure the responses, not just at one time, but the 3-hour time point, but at all of these different time points, to all of these different ligands. And I should say the platform for this is not 10x Genomics, it's BD Rhapsody, which has a much better technical noise profile. It's a targeted gene single-cell RNA-seq. We're looking at about 500 genes that are immune response genes in these macrophages. So the data in response to these different ligands and the different time points looks like this, right? And we have lots of data. But of course, unlike microscopy of NF-kappa B, where you're looking at the same cell in all the different microscopy frames, single-cell RNA-seq is a destructive technology. And so thereby, if we have different time points, 15 minutes, 30 minutes, 3 hours, and 8 hours, they're different cells. So how do I reconstruct a trajectory when I only have measurements from different cells? So I have to make an assumption about that there are cell archetypes that are giving rise to this data from which I'm sampling, right? So, so Catherine then developed and implemented an algorithm to infer the dynamic trajectories based on these time series data. And only when we have trajectory data, we can actually answer a simple question like, well, what is the fold induction? Because a question like fold induction means I have two time points from the same cell, the 0 hour and the stimulated hour, right? Or what is the integral of activation? I need multiple time points from the same cell. So single-cell RNA-seq alone doesn't allow you to answer that question. You have to infer the trajectories. And so the way she did that is to identify clusters using k-means through dimensionality, reduce the space, and then to use a spline interpolation of weighted by random walks to identify the simplest path that connects the different clusters and the different cells to infer trajectories in the data. So in other words, these disconnected measurements are then linked at the time points. And when you have time points linked, then of course you can infer the trajectory over the entire time course. And so, and therefore we have trajectories for CPG, for interferon beta. This particular gene, TNF, does not respond at all, and for these other ligands. And we can then ask the question, what is the variability of induction in response to the different ligands? And we see that some ligands are poorly distinguishable. You can see that the green and the red look very similar. So it makes sense that hierarchical clustering would mix them up, whereas other other ligands remain distinguishable. And that is now not done just for one gene, but for all genes, because we have 500 genes. And so here, 5 are shown. And of course, if you then have 5 genes, the clustering improves dramatically from doing that with just one gene. And so now for the first time, we have single cell RNA-seq trajectories, or single cell expression trajectories in macrophages in response to different ligands. And that's data that we never had available before. And so now we can ask, what are the features that correlate with ligands? Simple features like the peak amplitude, or the peak fold induction, or the integral of activation, or the speed of activation. And we can measure how much, for example, for this TNF gene, what are the features of in response to different stimulations, and what is the variability of that feature across hundreds of cells. And then using that, we can determine what is the information content in using Shannon information analysis. What is the information content of these features? And those are the last 4 columns here. The first 3 columns are time points. Here you see the 3-hour time point that I showed data for earlier. That's the information content for different genes. So some genes, the 3-hour time point shows a high amount of information, up to 1.5 bits. But most genes really don't show very good information content if you have a single measurement at 3 hours. Whereas if you look at integral, almost all genes show a high amount of information content if you have integral information available. So this just illustrates how much information we're losing by having only a single time point available. If we have more time points available that we're able to string together into a trajectory, there is an incredible amount of information in that data set. And so using these trajectories, or these features of the trajectories, we can now do clustering in a space defined by genes. So if we're considering just the peak amplitude, and for M0 macrophages, we see that these 3 genes are sufficient to distinguish different stimulation conditions. And fold change, not so well. Integral does very well also. And speed distinguishes some, but most of them not very well. So integral is probably the one that does best, and the mutual information calculation confirms that. And just to show that that actually changes with polarization. So the M0 macrophages, that's what I just showed. M1 macrophages, interferon gamma pre-stimulated macrophages, show an even better distinction of these. And you see how the variability is reduced in each case. So it indicates how M1 macrophages are more specialized and less heterogeneous in their responses. And again, this is not something that's evident from a single time point measurement. And here M2 macrophages are similarly more specialized and show a different distinction than the M0s. So my last conclusion then is that single cell gene expression trajectories can be inferred from single cell RNA-seq time series data, and that the dynamical features of gene expression are much more informative about cell states than single time points. And so I'm hoping that that is beginning to address what I promised, which is that cell identity may be defined through the network dynamics, not just the abundance measurements. And if there are challenges with determining network dynamics, but if those challenges can be overcome, they are likely to be more informative. So with that, I'd like to really highlight the 3 who I just realized are all MD PhD students. Ade Adelaya, Apeksha, who's gone on and is in his residency program, Apeksha Singh, who's still doing her PhD, and Catherine Xu is about to go to residency. So, and of course, the rest of my lab has also made great contributions to all of these. And thank you for your attention.
Really exciting. I think I'll check if there's any questions in the chat, and then we have a microphone here if you could use that for questions so that folks online can also hear you. But I'll kick off with a quick question, Alex.
Yeah.
We focused on NFKB signaling a lot in the context of macrophages, but I'm just wondering how many genes do you think are integrating that much different information through these sort of signaling codons? How ubiquitous is that?
How many genes are affecting the signaling codons or are downstream of the signaling codons?
I guess how many genes can create such a high dimensional response space with this sort of signaling codon approach? Is this like a lot of different genes do it, or just a few select genes in the human genome are playing this key role to integrate that much different information? I think,
well, so in the macrophage response, there's probably about 300 genes that are clearly NFKB responsive in the short term. And I think in some of this work, it appears that some genes rely more on a specific signaling codon and others on a different signaling codon. Now, in the end, of course, they probably they use everything. But you can identify, if you make perturbations to a particular signaling codon, or you look at correlations between signaling codon and gene expression, that certain genes predominate in what they use. The flip side, I think, that maybe that's what you were asking, is how many genes actually affect signaling, are participating in the signal transduction of NF-κB. And that's also in the order of hundreds. But we haven't done that. We haven't tried to correlate the expression of those regulators with the dynamics, with the dynamics of NF-κB. Roy Woolman's lab has attempted to do that in the case of calcium signaling, and that actually showed that the known regulators for calcium signaling do not very well predict the actual signaling dynamics of calcium. Yeah, there's more information somewhere else. So.
Yeah, so in follow-up Hannah's question, so let's say you compare the 7 or 8 different stimulus signals, right? Like including interferon, poly-IC, or the, let's say, okay, if all of them can stimulate the NF-κB pathway, but can you see the difference in terms of the downstream genes? Even if,
yes.
Yeah.
Absolutely.
Absolutely.
Yeah.
So even if they all can stimulate the NF-κB pathway, but the extent will be variable. Right?
Absolutely, yeah.
One is stronger than the other, which
right.
can be reflected by the downstream gene expression pattern.
Yes. Yeah.
Okay.
Yeah. So that's when I didn't go into that in this talk, but there's a whole bunch of papers that are from my lab and other labs that have attempted to address the mechanisms by which NF-κB target genes distinguish between these different dynamical features, and yeah, absolutely, they relate. Yeah.
Okay, and then the second question is that, we know that poly-IC can induce the interferon signaling. So basically, I think poly-IC will cover all the interferon stimulated genes.
Yes.
But in addition to that, can you see specifically what the other pathway that can be activated by poly-IC but not by interferon?
Yeah. Yeah. So you're right. That's makes it very, very, very challenging. So in addition to the dynamical code, there's a combinatorial code where multiple transcription factors work together. And that remains really an active area of research. So what we've done so far is we've knocked out the entire interferon system in order to study only the control of NF-κB and gene expression, and thereby could learn something about how dynamics control gene expression. But in a normal setting where the interferon pathway is present and the MAP kinase pathway is present, then we have to, we first will have to have information about the dynamics of each of these transcription factors in the same cell, and then gene expression of that. So you can see the technical challenges are there. There's still work to be done.
Okay, good.
Yeah.
Thanks.
So when you are analyzing the stimulus, I'm assuming you are doing it in vivo, not in vitro.
These cells are in vitro. I'm sorry. Yeah, all of those cells are in a dish, and they're stimulated in a dish by stimuli. Yeah, but they're live cells.
Right. But my first question is, if you're doing in vivo, do you think we will be able to detect them, because it's much more complex?
Yeah.
And both in vivo and in vitro, how many cells do you think we need to be able to detect the difference in these dynamics?
Yeah, remains a daunting challenge on how to understand these signaling dynamics in vivo, right? Because first of all, our measurement approaches make it very difficult to measure the dynamics in vivo. But then also the signals are much more complex. And, in vitro, we have, for example, we have a defined time zero. We know when we add the stimulus, right? In vivo, one stimulus goes to the next one, goes to the next one. So these studies really are sort of really just trying to understand the principles. And hopefully we're learning something about the principles, and then we can try to apply them to in vivo. But how to do that is certainly an area that people are thinking about, but in my lab, we haven't made progress. Yeah.
Great. Well, Alex, I posted something in the chat, but can you hear me? It's Ruben speaking here.
So sorry if it was actually mentioned during the talk, but problem is that, so why do you think the integral is like the most informative dynamical feature?
Yeah, why is that? Yeah, so well, it's in some sense it's the one that you would probably bet on because that determines how much protein is being made, right? So it's actually really sort of gratifying in some way that it's not something really complex, like speed. Like, why would speed be so important? But integral determines how much protein is made because that it tells you, how much mRNA has been there completely, and therefore the translation rate and the protein. So why is that the most informative in these studies? Maybe it is most resistant to cell-to-cell heterogeneity, that's sort of random fluctuations. Speed might be more variable, but then whether you activate at 10 minutes or 30 minutes, that would give you very different speed values. But then the response takes its course over time. It goes up, comes back down at a certain time, and maybe then the integral is more constant, which means it could be more stimulus specific, right? If the distribution to a single stimulus is more narrow. But we haven't looked into it. Personally, I've found it gratifying that it's integral because I can understand that biologically.
So I had a question
Well-
about the single cell seq trajectory reconstruction.
Yeah.
So presumably you have NF-kappa B data, maybe you have other data from other genes. I guess how well does it work compared to this actual single cell data? And then along with that, it seemed like there was actually quite a bit of heterogeneity between different cells in the actual single cell data for NF-kappa G trajectories.
Yes.
I imagine that's sort of washed out when you do the estimation.
Yes.
So how well does it work, and what do you think about all that extra variability that's ignored?
Well, the variability in you mean the single cell microscopy data, right?
Yeah, the microscopy.
The microscopy data, yeah, it was really daunting, it's been really daunting, which is why we went through these lengths of trying to identify features that might actually be correlated with ligands. It wasn't clear to begin with, other than that sort of oscillatory, non-oscillatory pattern. So the fact that we've found features that are indeed correlated through the statistical test was exciting, because I think the field was a little bit stuck, given how variable these trajectories are generally. But what was your first question?
So the question was when you did the single cell, you had the single cell seq, and then you reconstructed the trajectory, you estimated the trajectories. I'm just wondering how well that matches for genes that you might have it, like for NF-kappa B, how well that matches the actual trajectories. How well does it work?
Well, we don't have a gold standard. So when the microscopy for NF-kappa B trajectories is nuclear translocation. So it's not an expression level. It's just the same amount of protein. It just goes in the nucleus or it's in the cytoplasm. So we don't have gold standards. We would have gold standards if we do, for example, MS2 labeling. There's some fancy techniques by which you can measure RNA in real time, and that probably one should invest in that, but it's one gene at a time. Yeah.
I guess following up to some degree on that idea of gold standards, you mentioned the Woolman lab, who's sort of combining their calcium imaging with the spatial transcriptomic approach. And have you, I mean, I don't know what's experimentally possible, but have you considered combining your sort of live imaging of NF-kappa B with that sort of spatial transcriptomic information?
Yes, absolutely. In fact, we are doing that. So you would then, the nice thing about that is you can image NF-kappa B in a set of cells, and then that same cell examine target gene expression, right?
Right.
And so the question of combinatorial signaling came up, and so that's what So in that project, that's what we're attempting to do. We're measuring MAP kinase and NF-κB together, and then try to figure out which genes can be shown to correlate with a combination of those two signals. Yeah, it's a very powerful approach, and that's ongoing. It's a great, it does need good people. So if you're interested in that, be my guest. So it's a really great, really great project.
Okay. And then I guess second question, in your conclusions for your second part of your presentation, you kind of mentioned that different ligands distinguish or different ligand responses distinguish the polarization state better than others.
Yep.
But to some degree, shouldn't the cell be more informed of the polarization state already? In which case that would sort of indicate variability in response based off of the polarization state.
The polarization state puts the cell in a different steady state.
Right.
The question is, how do you measure that steady state, right? And so one way to measure the steady state is to measure the abundances of all proteins, which we can't do at a single cell, or of all RNAs, which we can do by single cell RNA-seq. So that's established. Now the question is, if we just measure abundances, are we missing something? Or is in the network dynamics, is there information there? And I can infer network dynamics by making the perturbation, by stimulating, right?
Right.
But maybe I missed the question. Maybe I missed what you were trying to get at.
Yeah. I guess like from a cell perspective, the cell's sort of like information environment, the polarization state, I mean, I could be wrong, but should be readily accessible to the cell in terms of information.
Yeah.
And so then, I mean, maybe it's just semantic, but then differences in response to different ligand signals being distinguished, or the responses that are well distinguished between different polarization states. Likely, it's not sort of informing the cell what its state is, but rather
Right.
the response it should be taking to some degree.
Yeah, the response to the stimulus is only informing us what the cell state is.
Right, right.
The cell state has a dynamic, but we don't know how to measure that.
Okay.
That's how I'm thinking about it. But
Okay. Yeah, it's probably like a little too semantic and yeah.
Well,
thanks.
maybe we will chat a little bit afterwards. Yeah, great.
Great, thanks. I think we're probably getting close to where we need to wrap up. You want to just check quickly if there's anything in the
There
chat?
are I don't know how to access the questions on is there a mouse for it's not on it's on this computer. But I'm sorry, I don't know how to get at the questions.
Can you hear us from the outside?
So please email me with your questions. Thank you very much, everyone. Okay, great. Thanks.
Thanks, everyone.